All right, so let's get started with class today. Um, Today we're going to be starting some lectures on the really hardcore molecular biology, stuff that you're going to be getting in MolCell. We're going to start with DNA and talk about the structure and function of DNA, deoxyribonucleic acid, which is the um, holder of all genetic information. Um, but we know that now. At the beginning of the 20th century, that wasn't known or understood. Um, it may seem like a well duh for us, but to the people in the 1920s, 1930s, it was a real interesting question. Where is the genetic material? So let me, there we go. Um, so scientists who were studying the genetic material, and this dates back to the experiments of Gregor Mendel with his pea plants, where he showed that information about the physical appearance of the pea plants, whether the peas were wrinkled or smooth, whether the plants were big or small, was contained in some kind of material that was passed from parent to offspring. And as more and more scientists became interested in the nature of this genetic material, some of the necessary properties of the genetic material became quite self-evident, right? Something that contains the information to make an entire tree or an entire human being must have a tremendous amount of information stored in it. So whatever the genetic information is, it has to be, or the genetic material is, I'm sorry, it has to be able to store information, very large amounts of information. All the information to develop from a fertilized egg to a complete organism, all the information about the structure and growth of that organism, and all the information about the metabolism of that organism. That's a tremendous amount of information. So the genetic material must be able to store large amounts of information. The genetic material must be replicable with a very high fidelity, right? You can't imagine having too many mistakes and passing it on to the next generation and having a successful next generation. So you had to be able to replicate it so you could pass it on to your offspring, but you had to be able to do so with a very high degree of confidence that you didn't make too many errors. But at the same time, it had to be mutable. It had to be changeable, because we know from evolutionary studies that changes in the genetic material allow for incremental changes in the properties of an organism. And that's how we get different species, different animals, different types of animals, are slow, gradual changes in the genetic material. So any substance within the cell that purports to be the genetic material has to meet these three criteria. It has to store information, it has to be replicable, but it also has to be mutable. Okay? And for the next few slides, I want to tell you a little bit about the story of some of the scientists who embarked on that quest to figure out what is the genetic material. So some of the earliest work that was done on the nature of the genetic material came from this guy, Frederick Griffith. In 1931, Griffith was experimenting with bacterial strains. And these were the strains that caused pneumonia. And there were two types of two strains of the same bacteria. One that was called the S strain, and they're pictured here. And when the S
Sorry
strain,
to interrupt, sir. I don't know if you have the slides up, but they're not showing for me at least.
oh really? Oh dear.
Correct.
Okay. David, what are you seeing? Just,
I just see your picture.
Same here.
Yeah, same here.
Yeah, you brought it up.
okay. All right, well, I'm an idiot. Yeah, I forgot to share my screen with you guys. That was my problem. Okay, let's go back. Sorry. All right. So this is what I was talking about, and hopefully you guys can see it now. Definitely, raise a hand if you can't see it now. But yes. So the properties of the genetic material, right? It had to store information, very large amounts of information about how to make an organism. It had to be replicable, and it had to be replicable with a high fidelity, right? You had to be able to make a pretty exact copy in order to pass your information to the next generation. But at the same time, it had to be mutable, changeable, to allow for incremental changes in the properties of an organism. So these were the three main properties that whatever molecule it turned out was the genetic material, which, around the turn of the 19th, 20th centuries, nobody really knew. It had to have those properties. And so the scientists who were investigating this had to keep that in mind as they were looking at different large molecules that we knew were present in the cell. Okay, so some of the earliest work was done by this guy, Frederick Griffith. And in 1931, he was experimenting with a strain of bacteria that caused pneumonia in laboratory mice. There are actually two strains of this same bacterial species. One was called the S strain and one was called the R strain. The S strain had a capsule around it that you could see under a microscope. The R strain did not have a capsule. When live S strain was injected into mice, they got sick and died. But when live R strain was injected into the mice, they didn't die. They survived the infection. Griffith also showed that when you took the S strain and you heated it and you killed it, and then you injected it into the mice, they survived. However, another experiment they did came up with a very interesting result. He took the S strain and heat killed it and mixed it with some live R strain. Now, normally those two conditions would allow the mice to survive, but when he mixed the two and then injected it, the mice died. And not only that, but when he tested their blood, he got live S strain back, where he'd never put live S strain in to begin with. And so what he concluded from these experiments is that bacteria can pass information about their virulence, how deadly they are, from one strain to another. And it was something that did not require one strain to be living. They could be dead, destroyed by heat, and yet something that they left behind was taken up by the live R strain, and it turned them deadly. And Griffith called that the transforming principle. And the molecular nature of that was something that people were very interested in. So about 13 years later, in the middle of the Second World War, this guy, Oswald Avery, did some experiments using the same strains that Griffith did, the S strain and the R strain. And he concluded that the transforming principle was DNA. And this is how he did it. He took the heat-killed S strain, right? And we know from Griffith's experiments that if you put heat-killed S strain and R strain together, then you get live S strain. You kill the mice. But what Griffith did is before he mixed them, he took the dead S strain and treated them with various enzymes. The first experiment he treated with proteases. These are enzymes that destroy proteins. When he mixed them, the S strain still became virulent, still killed the mice, and he still got live S strain out. So the proteases don't destroy the transforming principle, whatever it is. Then he mixed it with RNases, which destroy RNA. They didn't stop the transforming principle either. But when he treated the heat-killed S strain with DNases, enzymes that destroy DNA, that did stop the transformation. the mice survived, and he could no longer get live S strain back out of the affected mice. So he systematically tested the 3 large molecules that most people thought were likely to be the genetic material. And by process of elimination, not protein, not RNA, probably DNA, came to the conclusion that the transforming principle was DNA. And it had some of the hallmarks of what the genetic material should have. That is, DNA is a large molecule. It has a high molecular weight. And so it should be able to carry a significant amount of information encoded into that molecule somehow, encoded into the structure of the molecule. So Griffith concluded the transforming principle was DNA. But scientists, like everybody else, are human beings, and sometimes they remain irrationally attached to ideas that they've had for a long time. And there were many scientists who were convinced that not DNA, but protein was the genetic material. Proteins were also large molecules. It was known that proteins were much more diverse than DNA. DNA was made up of 4 nucleotides. Proteins were made up of 20 amino acids. And so people argued, if you wanted to carry a lot of information, wouldn't a more diverse molecule be your natural candidate? So there was still some debate, even after Avery's experiments, about what the transforming principle was. And it took experiments using bacteriophage, which are viruses that infect bacteria, to answer the question once and for all. Bacteriophage are really interesting little critters. They're viruses, just like the coronavirus is a virus, although that they have a very distinct structure, bacteriophage. They infect bacterial cells. And bacteriophage are basically a package of DNA surrounded by protein, and that's all they are. What they have to do in order to replicate themselves is find a bacterium, in this case E. coli, and inject their genetic material, and that causes the infected bacterial cell to produce more virus particles, and eventually the bacterial cell explodes, and all the little virus particles go off to infect the next bacterium. Okay. Now this is an electron micrograph of bacteriophage infecting a bacterium. This was not information that was available in the 1940s. But what you can see is here's the protein coat, and this dark material in the middle is the DNA. And we can see here, this one has not yet injected its DNA into the host bacterium, which is down here, but this one has, and it's empty. And here's one that's empty and it's leaving, it's been detached. But that made them the ideal experimental substrate because they were only DNA and protein. So if you could find out what part of the bacteriophage virus particle actually went into the DNA, you would have your answer. And you really only had 2 choices, DNA or protein. There was nothing else in the virus particle at all. And so this gave rise to a very famous experiment, which was called the Waring blender experiment, also called the Hershey-Chase experiment, after Alfred Hershey and Martha Chase, who actually did this experiment. This is a picture of them at Cold Spring Harbor Laboratories in the late 1950s, I think. But basically, they did this experiment with bacteriophage, and here's what they did. So I know this slide is a little bit complicated, but realize that there are really 2 separate experiments here. The one in pink on the top, going left to right, and the one in blue on the bottom, also going left to right. So after the Second World War, after the Manhattan Project and the atomic bombs, radioactive isotopes of common atoms were being generated in laboratories. And Hershey and Chase took advantage of that. They took radioactive sulfur, which only occurs in protein, and radioactive phosphorus, which only occurs in DNA. And they created either radioactive bacteriophage where the radioactivity was in the protein code, the pink guys up here, and not in the DNA. And they created radioactive bacteriophage where the radioactivity was in the DNA and not in the protein code, these blue guys down here. Then they took both of their creations and they tried to infect naive bacterial cells. But what they needed to do was make sure that once the cells were infected, they were able to separate the viral particles from the bacterial particles, because they figured whatever was infecting the bacteria, when it was injected, it was in the bacterium and no longer in the virus. But they had to do that in such a way that they had to agitate the solution that they were in such a way that it was gentle enough that it didn't break the bacterial cells open, because that would kill them and end the experiment. But it was brisk enough to shake these virus particles loose from the surface of the cell. And it turned out that the ideal machine was a Waring blender set on low. So what they did was they took this whole gamish and they put it in a Waring blender and they ran it on low. And then they centrifuged it. They spun it in a centrifuge at many times Earth's gravity. And so things separated out by weight. The bacteria, which were big and heavy, went to the bottom in what's called the pellet. The bacteriophage, which were much lighter, remained in the solution, in the liquid phase at the top. And then Hershey and Chase said, where is the radioactivity? Is it with the bacteriophage, where it was originally? Or is it with the bacteria in the pellet? And what they saw was that with the radioactive protein, the radioactivity remained with the bacteriophage in the liquid phase. But with the radioactive DNA, the radioactivity pelleted with the bacteria and did not remain with the virus particles. And so that was pretty much definitive proof that what the viruses had injected into the cell in order to replicate themselves was DNA and not protein. And that pretty much put an end to the protein people. They either gave up or nobody listened to them anymore, and they were stuck in their offices complaining to anyone who would listen about young punks and their radioactive isotopes. But this was pretty much the definitive proof that the transforming principle was DNA. So the next question became, okay, if the transforming principle is DNA, how does it carry so much information about how to make an entire organism? How can it be replicated so well that parents pass it on to offspring and the offspring are almost exactly the same as the parents? But how could it be changeable such that mutations can occur and populations can evolve over time? So that was the next question, the study of the structure of DNA. Now, this is a picture of a fragment of DNA. We know that DNA is composed of two main components: a sugar phosphate backbone. This is a deoxyribose sugar connected to a phosphate group. And the phosphate group is connected to the next sugar in line. And so they form a chain. And attached to these sugars are one of four nitrogenous bases. And that was already known. The DNA was composed of four nitrogenous bases: thymine, adenine, cytosine, and guanine. Furthermore, it was understood that DNA obeyed what was called Chargaff's rule. Erwin Chargaff was a former physicist who, after the Second World War, became very interested in DNA. And he looked all over and took samples of anything living that he could find: bacteria, animal, plant, virus, whatever, and looked at their DNA. he looked at the chemistry of the DNA. And one thing he found that was very interesting was that no matter what source of DNA he looked at, the proportion of the two bases was always in a 1-to-1 ratio. That is, however much A, adenine, was in the DNA, there was exactly that same amount of thymine, or T. And however much G, or guanine, was in the DNA, there was exactly the same amount of C, or cytosine. And this became called Chargaff's rule because he kept seeing it in sample after sample, plant, animal, bacteria, you name it. A was equal to T and G was equal to C. And so it became apparent that whatever structure DNA actually had, it had to explain Chargaff's rule, why he saw this. So this gets us to Watson and Crick, which of course everybody is familiar with, right? The famous model of DNA, the famous discovery of DNA that Watson and Crick came up with. So Watson and Crick were, Watson was American, Crick was an Englishman. They were working at Cambridge University in the UK in the late 1940s, early 1950s. And they were interested in the structure of DNA. They were using a technique called X-ray diffraction. And that was a technique for determining the structure of molecules. What you did was you took a molecule like DNA and you had to crystallize it. You had to remove all the water because water would mess with the crystal with the structure. And then when you did that, you zapped it with an X-ray, and the X-rays would bounce off the molecules in the DNA, and they would form a pattern on a piece of X-ray film. And if you knew what to look for, the pattern of blobs on the X-ray film told you something about the structure of the molecule that they were bouncing off of. So Watson and Crick looked at X-ray diffraction data, and they saw that from the X-ray diffraction data, it suggested that DNA was in the form of a double helix. That is, two strands of DNA wound around each other in a helical fashion. That is, they spun around each other, right? Like those little round staircases, except there were two of them. So Watson and Crick hypothesized that there was a mechanism, or there was a structure whereby the nitrogenous bases, one was attached to one strand and one was attached to the other strand, and they met in the middle of the DNA molecule. And furthermore, they saw that looking at the biochemistry of the four nucleotides, that if you put adenine and thymine in close proximity, they would make loose hydrogen bonds with each other. They would make two of them, actually. That is, they'd share a hydrogen nucleus. And guanine and cytosine would do the same thing, but they would make 3 hydrogen bonds. And as soon as they saw this in their model, they realized that this explained Chargaff's rule, that if A paired with T, one on each strand, and G paired with C, one on each strand, they would always have to be equal in amount. Furthermore, the Watson and Crick model implied a mechanism for replication. If you take the two strands apart and this pairing rule holds, then if I have a strand with just a T, I know that I have to add an A to it. And if I have a strand with just a C, I know I have to add a G to it. And so this was a way that DNA could be replicated with high fidelity and carry a large amount of information. Watson and Crick were competing, sort of, with another research group at University College London that was Maurice Wilkins and Rosalind Franklin. You may have heard the story about Watson and Crick and Wilkins and Franklin, and how Franklin got screwed over, basically. As a woman in science in the 1950s, she had it very hard. She was an expert crystallographer, and so a lot of the X-ray crystallography data that Watson and Crick ended up using came from her. But they weren't entirely competitive. In fact, they shared a lot of their data. Wilkins showed the X-ray diffraction data from Franklin to Watson, although Wilkins didn't ask Franklin's permission, he was also her boss. And Watson apparently had no knowledge of where the diffraction data came from. He just knew that when he saw it, it suggested to him that their model, Watson and Crick's model, was right. So Watson and Crick went ahead and wrote up their findings and published. They notified Wilkins and Franklin of their discovery, and Wilkins and Franklin published their findings in the same issue of the journal Nature right after Watson and Crick's paper. Ultimately, Watson, Crick, and Wilkins won the Nobel Prize. Franklin did not win the Nobel Prize, but not because she was deliberately excluded. It's because, unfortunately, she had passed away. She died a few years after this discovery of ovarian cancer. And the one thing that the Nobel Prizes do not do is award prizes posthumously. So because Franklin had passed away, she wasn't eligible for the Nobel Prize. There's no doubt that Watson and Crick are assholes, and that Franklin got screwed over. But it's a little bit more complex than many people portray it. But regardless, this is the model of DNA that we still use today. And really, there's no evidence that there's anything wrong about the way Watson and Crick perceived the structure to be. So, but there was another question, right? Every time you make a discovery in science, it raises another question. And this is no different. So Watson and Crick had shown a mechanism for replication. Take the two strands apart, and then for every base pair on one strand, you put the matching one from the other strand. So if it's an A, you put a T. If it's a T, you put an A. If it's a G, you put a C. If it's a C, you put a G. And that way you can replicate the two strands and make two copies of what was originally one strand. But how is it replicated? And the people who were studying DNA replication realized that there were three possible ways that a strand of DNA could be replicated during mitosis, during cell division. The first one is what's called the conservative model. Here's the parent cell in this column here. Here is the DNA of the first generation, so the two cells that are formed from the first parent cell's mitotic division. And here's the second generation, the four offspring that are composed of the mitotic division of each of the first offspring. In the conservative model, all of the parental DNA stays with one of the offspring. The other offspring all get new DNA. So they get the copy, right? It's like this is the original. The original gets kept with one individual and the other one gets the photocopy. Okay? And they pass on that copy. When the one with the original divides, again, one of its offspring gets all of the original DNA and one gets another copy. That's called the conservative model. A second model, which was also consistent with the data, was the semi-conservative model, where each of the two offspring of the original parent cell got one old and one new strand. And then in the second generation, one offspring got the old strand and the other one got completely new DNA. So in this case, the old strands were separated. They were shared between two of the offspring. And finally, there was technically a third possibility, the dispersive model, which basically said that the old and new DNA were all mixed up. Like it got chopped up and somehow pieced back together, and each daughter cell here in the first generation got some of the old and some of the new. And the same thing in the second generation, right? Each daughter cell got some of the old and some of the new. Right? The amount of old would be decreasing every generation, but everybody would get a little bit of it. This didn't make much sense in terms of fidelity, right? Keeping the genetic information the same from parent to offspring, but it was a theoretical possibility, so it had to be considered. None of these, all of these could not be right. right? They're mutually exclusive. Only one of them can be right. And this is an interesting insight into how science is done at an experimental level. When you think about a scientific experiment, when you think about doing a scientific experiment, you're trying to answer a question. And what you want to be able to do is you want to be able to, no matter what the experiment results are, get an answer to that question. Usually, it's of the type of answer either A or not A, right? So I might do an experiment and ask, is DNA replicated by the conservative model or not? And the answer I would get would be either yes, it is, or no, it isn't. If you wanted to distinguish between three possibilities, that is a lot of work. You have to run three separate experiments, and you run the risk of the answers conflicting. A better experiment, a better designed experiment, might give you the answer between two choices. So you might design an experiment which says, if I get one result that suggests DNA is replicated by the conservative model, if I get a second result that suggests it's replicated by the semi-conservative model. That's a more efficient experiment, but experiments like these are harder to design because you have to figure out what result gives me this, what result gives me that, and what other possible results are there, and how can I fit those into my calculations, my hypothesis. The idea of figuring out which of these three models is correct gave rise to what a lot of people have called the most elegant experiment in the history of molecular biology. And that's called the Meselson-Stahl experiment. And the reason people call it elegant is simply this. It was one experiment that took the researchers less than a day to do that distinguished definitively between all three possibilities for DNA replication. And it showed only one was possible. And as somebody who's designed experiments for over 30 years, I have to say that to be able to design an experiment like that, to be able to conceive of an experiment like that and carry it through is remarkable. Just the way they figured it out and the way they carried it out, and to do it so quickly and to make such a fundamental discovery about the nature of the universe and the nature of life on Earth was really remarkable. So what they did, in short, was, again, they made use of isotopes, in this case, isotopes of nitrogen. Okay. So they used heavy nitrogen, which had 15 neutrons in its nucleus, and so-called light nitrogen, which had 14. And literally, light nitrogen was lighter than heavy nitrogen. So if you put it in a centrifuge and you spun it really, really fast, it would separate out in terms of molecular weight, with the lightest stuff being at the top, the heaviest stuff down at the bottom. And if there was a mixture of the two, it would wind up in the middle as a hybrid, half and half. So what Meselson and Stahl did was they started with bacteria that they grew in heavy nitrogen. So the DNA would all be heavy. Then they transferred these cells to light nitrogen, and they let them go through 2 rounds of division in the light nitrogen. And then they looked at the result. They looked at the banding pattern. And if you remember the 3 models, this is what you would expect to see. Was there a question? No? Okay. Yeah, don't hesitate to raise your hand, obviously, if there's a question. So if DNA replication was conservative, you would expect there to be only 2 bands, a heavy band and a light band, right? The heavy band would be one offspring who got all the old DNA. The other would be the new offspring that got all the light DNA. So you'd expect only 2 bands. You would expect that after both the first and second generation. That is not what they saw. If it was dispersive, then you would expect to see a hybrid band after the first generation. But you would only also see a hybrid band after the second generation because everybody's DNA was a mixture of heavy and light nitrogen. They didn't see that either. What they saw was a result which very clearly demonstrated that DNA was replicated in a semi-conservative fashion. That is, the original heavy chain was split between the two offspring, and each one had one heavy and one light chain. So the first generation, they had only one intermediate band. But after the first generation of light nitrogen, the next generation would have two completely light bands and two medium bands. And so they would see two bands in their centrifuge tube. And that's exactly what they saw. And so their experiment, which, like I said, was basically run in a day, distinguished between these three possibilities and conclusively showed that DNA was replicated in a semi-conservative way. The one strand, each daughter cell gets one strand of old DNA from the parent and one new strand of newly synthesized DNA after mitosis. So, and that was a pretty remarkable experiment. So, let me just sum up what we talked about so far, the important experiments in the history of DNA. So Griffith and Avery both suggested that the transforming principle was DNA. Hershey and Chase, the Waring blender experiment, showed conclusively that it's DNA and not protein. And so they really settled that question. Watson and Crick then showed the structure of DNA. DNA was double-stranded, and one strand served as a template to make a new strand. And then Measles and Stahl showed that the replication was semi-conservative. Each daughter cell gets one old strand and one brand new strand after mitosis. All right, so any questions about that? Okay, so, but even though Meselson and Stahl had showed that DNA replication was semi-conservative, how that happened, how DNA got replicated, what the machinery was that did this in the cell over and over and over again, right? Every time a cell in your body divides, this has to happen. Every time a bacterial cell in culture divides, this has to happen. And some bacterial cells can divide in as little as 10 minutes a generation. So this process not only has to be very robust, right? It has to be able to happen in a lot of different conditions. It has to be fast. So this is what we know about how DNA gets replicated. So here's the basic idea. This is what Meselson and Stahl showed. The parent has one pair, one double helix of DNA, and the nucleotides A and T fit together, and G and C fit together. During S phase of mitosis, right, when DNA is replicated, the strands separate, and a new strand is made on each template, the left hand and the right hand. So the left hand template is the old one, and the new one is on this side. On the other one, the right hand template is the old one, and the new one is on this side. And yet, if you look at the DNA sequence, the nucleotides are exactly the same. These are two identical copies of the parent molecule. But one strand of nucleotides is old, comes from the parent. One strand is new. It's made immediately before mitosis. So how does this new strand get made? Before I go into that, I have to talk a little bit about DNA structure, because it turns out it has a very significant effect on how DNA is replicated. So here is a DNA double helix. Here is the sugar phosphate backbone of one strand, here on the left in blue. And here are the nucleotide bases of that strand. And here's the other strand in green. Here's the sugar phosphate backbone, and here are the nucleotide bases. Early research showed that the two strands of DNA are what are called anti-parallel. Okay? So DNA has two different ends to it, right? Just like a string has two ends to it, but in DNA, the ends are different. One end is called the 5' end because it has a phosphate that is just sitting out there. There's no next DNA molecule. The other end is called the 3' end because there is no phosphate there. In fact, there's just an OH group hanging off of this carbon here. And when you look at a double helix, a double strand of DNA, what you see is that one strand is oriented 5' to 3' if you look in one direction, say top to bottom. The other one is oriented in the opposite, 3' to 5' direction. So for a double strand of DNA, the 5' end is at the opposite side of the strand for each side. So one side is 5' to 3', one side is 3' to 5'. The problem comes with the enzyme that is responsible for making new strands of DNA, and that enzyme is called DNA polymerase. When DNA polymerase was discovered, it was found that DNA polymerase only goes 5' to 3'. That is, it only goes one way in replicating DNA. And so for replicating one strand, it would have to go top to bottom. In replicating the other, it would have to go bottom to top. So the DNA polymerase molecules have to run in opposite directions during DNA replication. But remember, these guys are still attached together. So how can you have a polymerase molecule going in both directions when in fact both strands of the DNA are still part of the same molecule? Right? It's like trying to zip a zipper both ways. It doesn't work. Turns out that there has to be some very interesting biochemistry in order to make that work. So DNA replication, how DNA is replicated, can be broken up into a few different phases. The first phase is what's called unwinding. So DNA is very tightly packed up in the nucleus. I think I mentioned before that an average strand of DNA from a single cell, if you took it and stretched it out, and this would be a human DNA, a human cell, the DNA would stretch about 6 feet, about 2 meters across. And it's packed into a nucleus that's only about 10/1 millionths of a meter wide. So that strand of DNA is very tightly compacted in there. It's all twisted up. Like if you took a rubber band and you held one end and you twisted the other end really, really fast, it all kind of bundles up and turns into a little ball, right? DNA is a lot like that. It gets packed up very tightly. So the first thing that has to happen is this DNA has to be unpacked. And there's a molecule called topoisomerase that goes along and basically cuts one strand of DNA, swivels it around, and then sticks it back together. And slowly it undoes all the knots that the DNA is tied up in when it's packed into the nucleus, so that you get a nice smooth ladder of DNA. But at this point, the strands are still attached to each other. Now, if you want to make a copy of each strand, right, the copying mechanism has to get in the middle there somehow. So the next thing that has to happen is the DNA double helix has to unzip, literally like a zipper. And for that, there's a molecule called DNA helicase that basically goes along and unzips the DNA strand. Once the DNA is single-stranded, it has a very strong propensity to come back together because its pair is only a few millionths of a meter away. So there are all these proteins called single-strand binding proteins that stick to the single-stranded DNA and keep it single-stranded until it can be replicated. This one replicated this way. This one replicated this way. Okay. So during the unwinding, the topoisomerase unwinds and breaks up the knots in the compacted DNA. The helicase then unzips one strand from the other. And the single strand binding proteins prevent the two strands from coming back together, at least too quickly. Okay, so now we have two strands of DNA separated. Now we have to bring in the molecule DNA polymerase and make a copy of each strand. Right? But the DNA polymerase only goes one way, 5' to 3'. So it would go left to right on the top strand, but it would have to go right to left on the bottom strand. And if it did, this molecule of DNA polymerase would zip off to the right and be fine, going and making a copy of the top strand. But the DNA polymerase for the bottom strand would go along and then it would run into helicase, and then it would get stuck. So the bottom strand has to be replicated by a different mechanism from the top strand. And that's what we're going to talk about. So the leading strand, or what's called the 3', or the 3' strand is called the leading strand. And this is replicated in the same direction as the helicase. So as the helicase moves along, the DNA polymerase follows it. Okay, this can go as one smooth, uniform movement. As the helicase unwinds the DNA, this follows. And that's leading strand replication. The DNA polymerase is working in its preferred 5' to 3' direction, so is the helicase. And so they simply both move in unison. Leading strand replication is simple. Both molecules move in the same direction, helicase and DNA polymerase, and this makes one copy of the top strand. But if the bottom strand went the same way, right, it would keep bumping into the helicase. The bottom strand has to be replicated left to right, away from where the helicase is moving, away from what's called the replication fork, which is this piece of DNA right here. So that has to be, the bottom strand has to be replicated by a different mechanism. This is how the bottom strand gets replicated. The bottom strand is the 5' or the lagging strand. This is replicated in the opposite direction from the helicase. So in this case, the helicase is moving off away to the left, but the DNA polymerase only moves left to right. So as the helicase leaves and creates a single-stranded region, a little enzyme comes in and makes a short primer of RNA. And this is a very interesting evolutionary artifact. Why this primer is RNA and not DNA is not clear, why it evolved this way. But it's clear that this method of replication is very ancient, probably about 1 billion years old. The DNA polymerase then binds to this RNA primer and starts to create a copy in the preferred 5' to 3' orientation. But it only replicates a little bit, and then it runs into the previous RNA primer. So on the lagging strand, the replication is done bit by bit instead of in one continuous line like the leading strand. And these little bits are called Okazaki fragments, after the Japanese scientist who discovered them. Once one fragment has been replicated, the helicase has moved off, and now another region of single-stranded DNA is exposed. So another molecule of DNA polymerase comes on and makes a new Okazaki fragment until it hits the previous one and falls off. And this continues overall from right to left. Eventually, there's an enzyme that comes along and removes the RNA and replaces it with DNA, which seems like a very inefficient process, right? Why not just have a DNA primer to begin with? But always remember, right? Evolution doesn't care about being efficient. Evolution doesn't care about how easy something is. If it works, then evolution goes with it. And this works. It may be not optimally efficient, but it's good enough. It's good enough that you can reproduce yourself, you can pass your genes on to the next generation, and that's all evolution really cares about. So yes, this could be more intelligently designed, but it works. And so that's what we've got. A lot of people, and this is a little bit of an aside, so don't worry too much about this. A lot of people argue that this is a remnant of the earliest life on Earth, which probably used RNA as a molecule to carry genetic information and not DNA. There's an interesting, it's called the RNA world hypothesis. If you are interested in reading about it, go to the wiki page for the RNA world hypothesis. And it explains how people think, or at least one hypothesis of how life arose on Earth, initially using only RNA, and then the function of RNA got specialized. The information carrying part went to DNA, and the doing things part, the enzyme part, went to protein. And RNA wound up as just a middleman. But it's an interesting hypothesis. Although, like most speculation about the beginning of life on Earth, it's still only a hypothesis, and it's going to be very difficult to prove, right? It's very difficult to prove what happened 2-3 billion years ago on Earth, especially since DNA and RNA don't last that long. There's no such thing as fossil DNA. So here's the whole thing put together. Unfortunately, it goes in the opposite direction from the other figures that I showed you, okay? So here's the helicase, the blue guy. The topoisomerase isn't in this picture. It's already done its job. So here's the nice, smooth parental DNA helix, and helicase is going from left to right, unwinding it. The leading strand is up at the top. The DNA polymerase on the leading strand is moving in its preferred 5' to 3' direction, which follows the helicase. So as the helicase unwinds the DNA, the leading strand polymerase follows. It can do it all in one go. It doesn't have to stop. But in order to replicate the lagging strand, the DNA polymerase has to move backwards. And so as the helicase leaves this exposed piece of single-stranded DNA, an RNA primer gets set down, a DNA polymerase makes an Okazaki fragment, and then it lets go of the DNA strand. Meanwhile, the helicase has moved on, and so a new primer is put in place, and a new DNA polymerase molecule then continues until it runs into the old one. So while the leading strand is replicated in one continuous go along the entirety of the DNA molecule, the lagging strand is replicated in little blips, little bursts, little fragments that are eventually stitched together. All right, so I know this is a lot of information, and it's particularly difficult, at least for me, to gain an understanding of it from a static image. And so I have a movie of exactly this process to show you. It comes from the Howard Hughes Medical Institute, a very wealthy research institute. And I call it DNA Replication, the movie. Right? Aren't you terrified? Okay. So we'll see how this goes. You should be able to see the movie and hear the sound. If you don't see the video and hear the sound, please raise your hand and let me know. So cross your fingers. Here we go.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. It occurs in a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite 3' to 5' orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding protein, and in some systems, multiple Okazaki fragments may be present. The molecular reality is very different from the iconic image of the double helix neatly separating into two DNA copies, as so often depicted.
Okay, so I hope if the static images didn't make as much sense to you, hopefully that movie did. It's available at the Howard Hughes Medical Institute. I'll put up a link on the Moodle page for it. So one last slide. So don't despair. We are almost done. I just wanted to contrast DNA replication of prokaryotes, bacterial cells, versus eukaryotes, right, plant and animal cells. Prokaryotic chromosomes are pretty small usually, and they're usually circular. There's usually one chromosome and it's usually round. And so it gets replicated in both directions, starting at one origin. And both go halfway around and eventually produce two complete daughter strands. right? And each one, it's semi-conservative. Each one has a parent strand, and each one has, or an old strand, and each one has a new one. Now, since prokaryotic chromosomes are pretty small, they can be replicated very quickly. And like I said, some bacteria can divide and replicate their DNA and completely divide in less than 10 minutes, which is amazingly fast. On the bottom here, these are eukaryotic chromosomes. Eukaryotic chromosomes are much larger than prokaryotic chromosomes, many orders of magnitude larger. And they're linear. They're in straight lines. They're not in circles. So if eukaryotes used the same replication strategy, it would take much longer to replicate eukaryotic DNA. But that's not the case. Speed is critical in terms of replicating DNA. It's the rate limiting step for how fast your cells can divide. So what you see in eukaryotic DNA is more often than not, DNA replication occurs at multiple points along the chromosome at the same time. Each strand is replicated in both directions, creating what's called a replication bubble, right? One set of DNA polymerases is working on this side of the replication bubble, moving right to left. Another set of DNA polymerases is working on the right side of the replication bubble, moving left to right, outwards. And here's another one, same way. Sorry. Eventually, these replication bubbles will meet. And so the entire DNA strand will be replicated, but much faster than if one set of molecules just moved along the whole strand from one end to the other, right? It's done at multiple sites, initiated at multiple sites, all at the same time. So eukaryotic chromosomes are much larger, but are replicated fairly rapidly because multiple places on the chromosome are replicated at the same time. Whereas bacterial chromosomes, they start only at one place and go in both directions around the circle. Because bacterial chromosomes are a circle, they meet in the middle. Right? Here, eukaryotic chromosomes are linear. When they get to the end, the molecular machinery just falls off. And then it goes and finds another place in the DNA to replicate. Okay. So that is DNA replication. Hopefully you understand a little bit about the history of it, what experiments were done to show that DNA was in fact the transforming principle, how the structure of DNA suggests that it can be replicated, mutated, and yet store large amounts of information. Although we're not quite there in figuring out how the information is turned into cellular components. We'll talk about that on Friday. We talk about transcription and translation. So, any questions then about what we discussed today? DNA replication, DNA structure and function? Ryan, you have a question?
Yeah, not necessarily about the subject, but how thorough, in terms of like the processes and the pieces, do you want us to be about DNA replication for the exam?
Well, okay. Everything I've showed you, I think is important. I'd like you to know the major thrust of each of the experiments that we talked about. So what did Avery show? What did Griffith show? What did Hershey and Chase show? And why was it important? When we talk about the mechanism of DNA replication, I want you to understand how the DNA bases pair, A with T and G with C, how they're separated, how they're copied, know the differences between how the leading strand is copied and how the lagging strand is copied. And just know a little bit about how bacterial chromosomes are replicated versus eukaryotic chromosomes.
All right. Thank
Right?
you.
Okay. All right, Rob. Trey?
Yeah, so will our exam be set up like our previous ones, or is it going to be different because we are taking it at home?
So at this point, I do not plan to make the final exam any different than I would if you took it in class, except it will be on Moodle. I might have to tweak a couple of the questions so that you can answer them in a multiple choice format rather than drawing a diagram or something like I would normally do. But it'll still be, the first part will be true, false, fill in the blanks, multiple choice. There'll be a few short answer questions or maybe draw a diagram. And then there'll be one essay question. And there's only gonna be one essay question, and it will be the one on the exam. And I will let you see it a week in advance. So you have the option of writing up your complete essay and just copy pasting it during exam time. Because it's online, you are allowed to use your notes. I wouldn't recommend using the book, but obviously I'm not there to watch you. So you'll have the full 2 hours. The exam is timed, but you have the full 2 hours to take it. But it's no longer than any of the other 2 exams. So if you finished exam 1 and 2 in 45 minutes, you should probably finish this one in 45 minutes, if not sooner. Hopefully that answers your question, Trey. Yes?
Yeah, it did. Thank you.
Okay. Okay. Any more questions about the material or about the exam?
I have a question. You might have said it at the beginning of the class, but you said it was going to be posted on YouTube. Can you post the link to that video on Moodle or something when it's posted?
Yes, I will. So Zoom, once I'm done with the meeting, it will take a little while to process and create the video file. And then once I have it, once it's posted on YouTube, I'll put up the link on Moodle.
Thank you.
So yes, I'll live forever on YouTube. Maybe I'll go viral, who knows? All right. All right, so Friday we'll talk about transcription and translation. Until then, stay safe, stay sane. Hope you guys are doing okay despite the confinement. And yeah, I will talk to you on Friday. Okay, bye all.